Thanks for the introduction, Alexa. Good morning, everyone. I'm so glad to be able to join you guys. I'm often focused on the other end of these studies, so it's nice to talk to the people who are out there actually interacting with the participants and doing all the hard work, actually. I'm not muted, right? Oh, okay, good. Am I muted? So today I'm going to tell you a little bit about some of the studies Alexa just mentioned with a focus on March stress of COVID, because that's the new project, which you probably haven't heard as much about. But I didn't want to dive just right into that because I feel like it's always good to have some context and be reminded about what came before. So as Alexa mentioned, my group studies the bacterial communities of ARCH and MARCH participants. And our current focus is the impact of those microbiota communities on perinatal and infant outcomes. And I'm an assistant professor here at Michigan State University, working from home in my basement these days, but working for the university all the same. So what is the microbiota? These are these little microbes, bacteria, viruses, fungi that live actually in and on us. My lab focuses on those microbes which actually live in our gastrointestinal tract. The studies I'll talk about today are not only focused on the microbes that live in our gastrointestinal tracts, but also on those microbes that live in our vaginal tract. And that's mainly through a collaboration with Kim McKee at the University of Michigan. She's very interested in the vaginal microbiome and its relationship to perinatal outcomes. And when she heard about March and ECHO, she contacted us. And so now, in addition to looking at the gut microbes, we're often also looking at the vaginal microbes, which you'll see when I describe some of these studies to you. So why are we interested in these little microbes that live in and on us in our gastrointestinal tract and our vaginal canal? Well, we're really interested in them, one, because we don't know that much about them, although what we do know about them is growing by leaps and bounds every day. And we're really interested in them because this perinatal period, this time between when you're in utero and when you're born, is a key window of opportunity for us to be colonized with these microbes. And pretty much once we get colonized with these microbes and we're between 1 and 3 years of age, our little friends are with us, and it's going to be really hard for us to change them. And so this window of time during birth and infancy is really important for understanding exactly what's going on with these microbes. And you may be wondering, well, how do they actually interact with us? These microbes can metabolize nutrients that we're not using. So we eat food, and we know some of that food we don't use, it just comes right out of us. These microbes living in our GI tract can actually interact with some of those food components that we don't actually use, that our body can't act on. And so these microbes then, when they act on those food components, can create new molecules, new chemistry that can actually interact with not only our gastrointestinal tract, but our immune systems, our brains, and these other parts of our bodies. And so that's why we're really interested in these microbes that live in these mucosal environments. So we're interested in this pregnancy period. So what's happening with mom's microbiomes during pregnancy. And we're also interested in what's happening with the children's microbiomes as they're developing through 1 year of age, being exposed to pharmaceuticals, bacteria from their diet, environmental and familial exposures, in addition to having a pretty limited diet in infancy. As children go on to solid food introduction, their microbial communities change quite a bit. And so we like to follow these children through 1 to 3 years of age so we can get a sense sort of what their microbial community is going to look like later. And so just if we want to focus on the infant here, these little funny looking shapes inside the arrow are depictions of microbes. And the microbes actually kind of look like this, like they're rods or they're Y-shaped or they're little circles. We think that during gestation, some of mom's microbiota may actually influence gestational outcomes. And we know that from birth through toddlerhood, that these microbes, which are living in and on this child, can impact their development, their immune system development, their gastrointestinal tract development, and their neurodevelopment. But we are still trying to learn a lot of details about these interactions. So we have a general sense that these things are important, but we don't know a lot of those details. And so we use the longitudinal pregnancy and birth cohort to actually start to understand some of these details, in addition to confirming some of what's been reported before about the general trends of this relationship. So what are some of the knowledge gaps? So up here, I have pictures that depict the vaginal microbiome and the gastrointestinal tract microbiome, just to remind us that we're thinking about both of these things. So one knowledge gap is that we don't know the impact of the maternal, vaginal, and gut microbiota on perinatal outcomes, such as length of gestation, birth weight, or weight for gestational age. We also don't know all of the factors that determine the gut microbiota assembly. So that means how we get the different microbes and how they're coming together and living happily in us when they're being initially established during this window of opportunity, which is infancy. So up until now, these have been fairly generally poorly described, and we're trying to describe these in much more detail using longitudinal data. And that longitudinal data is important because without it, we can't really say if the microbes are preceding the outcomes. And so in this way, by studying the microbes longitudinally, we can kind of say whether or not the microbial composition, those little guys living in us or in the babies, are having an impact later on health outcomes. And then finally, it's unknown how the gut microbiota community in early life is related to infant as well as later life growth, development, and health. So we have some clues about what's going on, but again, as I mentioned, a lot of the details are completely unknown, and we're working hard at trying to understand these details. So the general model is that the maternal microbiota could impact these perinatal outcomes, such as length of gestation, birth weight, small for gestational age, but also the maternal microbiota could impact this child's gut bacterial community, that the child's gut bacterial community goes on then to impact those molecules, those chemicals that are being made by the bacteria, and that those molecules that the bacteria are making or that are associated with those bacteria will impact that child's growth and neurodevelopment and other types of development. So enter the studies. So Alexa alluded to this. So my first set of studies were based on 40 maternal infant dyads who were registered either into ArchGut or Baby Gut. And ArchGut should be well known to you because that's the Archive for Research on Child Health, of which we have 800 dyads actually, but I did not get involved in this project until 2015 and didn't start recruiting dyads until 2016. Or I may have recruited my first dyad at the very end of 2015. And so I only was able to pick up on those dyads who came in at the tail end of Arch. And as we know, Arch is mainly based in Lansing, but also has some dyads from Traverse City, although some people call the Traverse City dyads March Light or like pre-March. And then Baby Gut was completely focused in the greater Lansing area. And so for ArchGut and Baby Gut, we follow a lot of the same protocols that I'm going to be talking about for the new study, the March stress of COVID study. We consent women during pregnancy. They complete some enrollment questionnaires. During pregnancy, they collect an adult stool sample. Then they collect stool samples from their infants at 1 week, 6 months, 1 year, and 2 years of age. And then we keep following up with these dyads looking at growth and intestinal health every year up until 5 years of age. And right now, the majority of the children are the women who join this study. Their children are about 4 years old right now. So we've published several papers related to that cohort. One was looking at the association between pre-pregnancy BMI and the infant and pregnancy microbes that are in the gastrointestinal tract. Two was looking at how pregnancy dietary diversity was associated with pre-pregnancy BMI among women in the Michigan pregnancy cohort. And you might know this nice woman right here, Brianna Kornatowski. She was the first author on that paper when she was working in my lab. In collaboration with Dr. Zhang in the MMG and Epi and Biostats department, we used these samples to understand some of the perinatal risk factors that were related to antibiotic resistance. So these microbes that live within us can pick up genes from the environment, which may make them resistant to exposure to things like antibiotics. And so we were looking at that in this paper with Dr. Zhang. And then finally, we were looking at human milk and how it was related to the 6-month microbiome and whether or not maternal BMI was also associated with the 6-month microbiome. And I'm going to show you just a little bit of data. I know you're not really data people, but as a research scientist, I have a hard time talking to people without talking about some data. So one of the really interesting things that I like to show everyone is that we can actually track someone's microbial community. So each little dot here represents a single sample from a single person. And it represents the whole of their microbial community as it relates to the whole of all of these other people's microbial communities. And the further apart 2 dots are on this plot, either in this direction, so the X direction across your screen, or in the Y direction, so the up and down your screen, the further apart these 2 dots are, the more different the microbes are that are inside that person's gut community. So in black, we have pregnant women, and these are what their gut microbial communities look like. So when we're plotting the gut microbial communities of pregnant women on the same graph as the gut microbial communities of infants, all of these pregnant women have much more similar gut microbial communities to each other than they do, say, to these red dots over here, which are the microbial communities you find in a 1-week-old baby. And what you'll notice about the babies is that as they get older, from 1 week to 6 months to 12 months, their gut microbial communities are progressing and becoming more and more similar to that of their mothers, to that of those microbial communities of the pregnant women. So I think this is just a really striking demonstration of that maturation, or how our gut microbes change from birth through that first year of life. And so at 6 months of age, we got those poop samples from the babies. The moms collected them and sent them to us. And we did the same thing. We looked at how these gut microbial communities within just 6-month-olds right now were different from each other and what factors contributed to those differences. And I was really interested in how maternal pre-pregnancy obesity would impact those 6-month-old gut microbial communities. Well, it turned out when we plotted this data, in black, we have women who were babies from women who were normal weight before birth. In red, we have babies from women who were overweight before birth. And in green, we have women, babies from women who were obese before those babies were born. We're super excited because obesity seems to be explaining this variation in the gut microbial communities, because the green dots are sort of circulated over here, whereas the red and the black dots are circulated over here. However, when we looked a little closer, we realized that it was actually the women who were not breastfeeding very much whose babies had more similar gut microbial communities compared to the babies from women who were breastfeeding. And I'm going to show you some data which will demonstrate that this is related to mom's BMI. So on this plot, we have different shapes. The circles mean that baby actually did not have, half of that baby's diet was not human milk. And then the squares mean that greater than or equal to 50% of that baby's diet was human milk. And the squares, of course, are gathering together over here on the right-hand side of the screen, and the circles are gathering over here on the left-hand side of the screen, which the green dots mean that the mother was obese prior to birth. The black dots mean that the mother was normal weight prior to birth, and the red means that the moms were overweight prior to birth. And so you can see how the low rates of breastfeeding are really overlapping with those obese moms, and the high rates of breastfeeding are really overlapping with normal and overweight moms. So it's not really mom's pre-pregnancy BMI that's driving these differences in these babies' microbes. Rather, it's this exposure to human milk. And so we wanted to look at rates of breastfeeding amongst these women who were obese prior to pregnancy. And what we saw is that at 1 week of age, there's a little bit of a difference. So the black bars are moms who were normal weight. The red bars are moms who were overweight. And the open bars are moms who were obese prior to becoming pregnant. And this percentage on the left-hand side is the percent of those within those categories that were feeding milk from the breast, that were feeding milk from the bottle, and that were feeding formula. So while milk from the breast doesn't differ very much, what you will notice is that more overweight and obese moms were feeding some formula at 1 week of age. And as we progress, so now at 26 weeks, which is 6 months of age, normal weight moms are feeding much more milk from the breast compared to overweight and obese moms. And again, over here at 6 months of age, fewer of the normal weight moms are feeding formula, and more of the obese moms are feeding formula. And then this kind of falls apart at 1 year of age because very few people are breastfeeding at that time point. Okay. So again, just highlighting here at 1 week of age, almost all of the moms are trying to feed some breast milk. But at 1 week of age, many more of the obese and overweight moms are feeding formula. By 6 months of age, the number of obese and overweight moms who are feeding breast milk really drops off. And this is also true not just at the breast, but also giving their pumped milk in bottles. And the number of overweight and obese moms who are giving formula increases. And so what we can conclude from our early studies with arch gut and baby gut is that dietary intake of human milk is this really important factor in the infant microbiota. And it's associated with maternal pre-pregnancy BMI. And so that those microbial changes we thought might have been associated with BMI are really just reflective of those infants' diets. And also what we realized is that our sample size of 40 is really not big enough to enable us to really carefully categorize moms and babies by how much human milk they're getting. We'd really like to look at, is there a dose of human milk that can is enough basically on top of formula that can help give you the microbiome, which can set your immune system and your neurodevelopment on the right track. So enter the March study with which you guys are very instrumental in, being the recruiters out there, getting women to participate in this study. And now, instead of just being focused in Lansing and Traverse City, we're going to get dyads from all over the state. Well, all over the lower peninsula of the state. And so in the microbiota studies that are occurring in March, the main sample collection is at 3 months of age, where we're trying to get every March participant to send us a 3-month fecal sample from their baby, a 3-month poop sample from their baby. So you guys give them their 3-month questionnaire survey. And during that survey, you ask them if they'd be willing to donate a poop sample. And if they say yes, then SRC over at the University of Michigan sends those moms a sampling kit. And when those moms collect that poop, it comes to me in my lab, and we're using it to try to answer this question about how the extent of breast milk exposure influences these gut microbial communities and these babies. So thank you for helping us to get all those moms to send their 3-month samples to us. But we also have 3 sub-studies that are going on, March swab, March microbe, and March stress of COVID. And the March stress of COVID is the new study that we just started. And I'm going to tell you a little bit more about each of these studies just so that you have a better sense of what moms can sign up for if they're interested. So March swab is a single sample collection during pregnancy, and that sample collection is a vaginal swab. And taking this vaginal swab is very similar to inserting a tampon, so it's not something that moms are unfamiliar with. It's a very familiar process. And these are what the little swabs look like. They come in a little plastic container. Mom uses, we give her gloves actually, and she can then take the swabs out and without touching the stick part of this, insert it similar to a tampon, swirl it around the vagina, and then put those sample swabs back into the plastic tube, and then they mail them to us. And so this is really just one sample during the 3rd trimester of pregnancy for us to understand what that vaginal microbiome looks like in that time period before birth. And then we can use this information to try to understand the impact of the vaginal microbiome on some of those perinatal outcomes I talked about at the beginning, such as gestational age at birth, birth weight, and small for gestational age. And so these women, if they collect this sample, then they're incentivized with a $15 gift card. For March Microbe, what we ask of the moms is to donate a poop sample during their 3rd trimester of pregnancy, and then to collect a poop sample from their infant when that infant is around 1 month of age, and then to collect another poop sample from their baby when that baby is around 6 months of age. And of course, at each of these time points, they answer some questions about diet, medications, and growth of the babies. And so if mom sends us her poop sample, then she'll get a $20 gift card. And then for each infancy sample, she'll get a $10 gift card. All right, and then for this sample collection, we send mom a plastic tub, and this plastic tub just sits in your toilet. This is my toilet. And you put it between the porcelain and the seat, and then you can just sit down, and it happens to be located pretty much perfectly to collect your poop, but not your pee. And then we give mom some gloves so that she can use the little scooper that's in the lid of the tube here to scoop some of that poop and put it into this tube. For the baby, of course, babies don't sit on toilets. So instead we send diapers to the moms. And again, they can use the scooper that's in the lid to scoop the material from the diaper into the tube. And right now we're sending 2 different kinds of tubes. We're sending the white cap tube, which is basically an empty sterile tube. But we're also sending this purple cap tube, which has a preservative in it, which protects the DNA from the samples from degrading. That DNA is really important for us because that's what we're using to say what kind of microbes are in the sample. And so I just created this little handout that moms get, which show how to collect this sample. This tube comes in a sterile pouch with a little plastic spatula. You can use that plastic spatula there to just put a little bit of sample right on the top of this yellow part, which I've shown here. You put that purple cap back on. You want to shake it up and you want it to look mixed up in the end. I also said, I show the white tube here that then you're going to take some poop, put it in the white tube, screw the lid on. And this lid has a spoon built in, which is really nice. Then we ask that the moms put both tubes into a plastic bag with an absorbent material, which in this case is a paper towel. Seal that bag shut, put it in a secondary container, which is a Tyvek envelope. Shove that envelope into the box, complete some questionnaires, shove those into the box, close the box and drop it in the mail. And also in addition to dropping in the mail, which I'll show you later, you can request pickup by the US Postal Service. So now we have a new study, which is going to be kicking off any moment now, called March Stress of COVID. And this is one of the more involved studies. It includes 3 time points in pregnancy. And at each of these 3 time points, moms will collect a dried blood spot, collect a spit sample, collect a hair sample, and collect a vaginal swab sample. We're using the dried blood spots in collaboration with New York University Children's Hospital environmental study to look at antibodies to COVID in their blood samples. We're using the spit sample to look at RNA for COVID. We're using the hair samples to look at cortisol levels in mom. And we can use hair cortisol as a rough marker of stress. And then we're using the vaginal swabs pregnancy because this aligns more with what Kim McKee at University of Michigan truly wants to look at, which is sort of that temporal change in the vaginal microbiome during pregnancy and how it impacts the vaginal microbiome, or the perinatal outcomes, sorry. And then in this 3rd time point, which is the 3rd trimester, we're also asking moms to send us a poop sample. And so at each of these 3 time points, they'll be incentivized. The first 2 are $20 each, gift cards, and the last one is a $60 gift card. And then additionally, depending on the age of the woman's infant, we will be contacting women who have infants between 0 and 1 year of age to try to collect stool samples. If that infant is very young when they enroll in the study, we'll hopefully collect all 8 infant stool samples spread out over the course from birth to 1 year of age. If that infant is older, we'll just collect those fecal samples we can get from those infants. And so for every child stool sample or poop sample that a mom sends in, they'll get a $20 gift card. And what March Stress of COVID is trying to do is connect some of the stress that mom is experiencing during COVID, during her pregnancy, to those perinatal outcomes. And we think that that stress that she's experiencing can impact her microbial communities, either in her vaginal canal or in her gastrointestinal tract, and that can then impact those perinatal outcomes. And in the infant, we're interested in how maternal stress can impact the stool microbiome of these infants, but we'll also be looking at infant neurodevelopmental outcomes, which are measured at 9 months of age in the full study, to see if infants have different microbiomes during this time point of COVID, during our experience of COVID, and whether or not that's impacting their neurodevelopment. And then I did just want to show you guys that if a mom is shipping back her kits and she doesn't want to drop off at the post office, she can just drop it off in the mail bin found in the lobby of pretty much any US post office. But she can also request a pickup if she goes to USPS.com. At the very top of that webpage, there's a little bar, and if she clicks on send, she'll see this next window pop up, and she can click on schedule a pickup, and then she can set up a time for the Postal Service to pick up the package from her front porch or mailbox. Of course, this research is not done in a vacuum. None of this could be done without funding from CHARM and ECHO. None of this could be done without Dr. Paneth. I have other collaborators who've helped with breastfeeding and microbiome stuff, students who've been very important, 2 of which you guys might recognize the names of, Brianna Kornatowski and Kristen Schmidt. My lab now is a little bit bigger, although Cameron has graduated and Alyssa is now working at Wayne State as a faculty member. Some of these other students have also graduated, but still working with a lot of these students. And that's all. I'd be happy to take any questions.